Hello, I'm Dr. Kishore Kumar. It's a pleasure to talk to you today. I'll be talking on the genetics of dystonia, and this is for Dystonia Awareness Month. And I'd like to thank the Dystonia Network of Australia, particularly Robin, for asking me to talk. As a bit of an introduction, I work at Concord Hospital. I'm a neurologist there. I work on the neurology department and also in a genetic testing laboratory called the Molecular Medicine Laboratory. And I also work at the Garvan Institute as a researcher, and I lead a genomics research group. So today I've been asked to talk about our research into dystonia, our findings so far, results, developments, and discoveries we've made. So an important place to start is what causes dystonia. So, dystonia can be due to many different causes. It can be acquired, so it can be due to like a birth injury, cerebral palsy, infection, inflammation, drugs, toxins, stroke, brain tumors, and it can be due to a problem with the mind, functional or psychogenic. It can be due to genetic factors. And it can be due to um, we may never know the reason why a certain person has dystonia, and it can be due to a combination of these factors. But the part that interests me is the genetic cause of dystonia. And here we can divide it up. We can say you have a mutation in a single gene, and that has a strong effect and is the cause of your dystonia. And we call that single gene or monogenic. In comparison, dystonia could be due to many different genes, genetic risk factors, and this could be called polygenic or complex. So common variants in many genes can also contribute to dystonia. But my research is primarily focused on monogenic dystonia, which means a mutation in a single gene that has a rare mutation that has a strong effect that's really causing dystonia in that person. So, what I'm fascinated about is using advances in genomics to try to learn more about dystonia and other movement disorders and to give people a genetic diagnosis. And our first study looked at genome sequencing. So traditionally, we would have sequenced a small part of the DNA with Sanger sequencing, which is like first generation sequencing or targeted sequencing. Or, and then we moved on and we did second generation sequencing uh, using next generation sequencing panels where we were able to sequence a lot of DNA, sequence a large amount of DNA in a single experiment. Whole exome sequencing, where we sequence every gene in the body, all 20,000 genes, and genome sequencing, where we sequence all the genes, all 20,000 genes, and all the spaces between the genes, the non-coding regions. And my first studies into dystonia focused on whole genome sequencing. And this was our paper that we published in 2019. And we really were using that technology, which was really advanced at the time, called whole genome sequencing, and applied it to patients with dystonia. And we took patients who were just referred to us. We weren't very selective. We took over 100 patients and we did genome sequencing on them. And this was really one of the first studies in the world to study dedicated to genome sequencing in dystonia. So we studied 111 patients with different types of dystonia, and we found a genetic diagnosis in 11.7%, or 13 out of the 111 individuals. We used a different approach. So if you do genome sequencing, you get a vast amount of genomic data back, many different changes in the DNA, many different variants. And the trick is to figure out if one of those variants is the cause of dystonia or the cause of a disease. It's like trying to find a needle in a haystack. One way we looked at genes that we already know cause dystonia. This is a list of genes that we looked at, and we went through the genome data and tried to pick out a mutation in one of these genes. So these are the different genes that we identified: ADCY5, ATM, GNAL, KMT2B, Parkin, epsilon sarcoglycan or SGCE, and THAP1. So these are names of genes that we already know linked with dystonia. It seems like a modest diagnostic rate of 11%, but keep in mind that most patients with dystonia don't get genetic testing, and trying to find a cause of disorders, a genetic cause in disorders like Parkinson's and dystonia is very tough. Here's an example of a patient who we did diagnose, a 42-year-old woman whose symptoms began at 32 years. She had problems speaking. She became mute on the telephone or when she's anxious. She noticed a jerky quality of her voice, and she developed neck dystonia with her head being pulled back, and she had twitching of her eyes. And a key point is that she had a family history with her mother having spasms around her nose and eyes, and her mother's paternal grandfather also having a voice condition. We thought the voice condition could have been spasmodic dysphonia. This is what we call a pedigree or family tree. You can see the patient here indicated with the arrow, the affected mother, and the paternal grandfather of the mother. Uh, People who are affected are filled in black. And here, using genome sequencing, we identified a mutation in the GNAL gene. And here is the patient here. And you can see Professor Sue, Carolyn Sue is examining the patient. You can see her voice is got this strained quality from spasmodic dysphonia, and her head is being pulled back. Cervical dystonia.
Today is a sunny day today.
Also, using our genome sequencing technique, we're able to look very carefully for hard to find mutations, such as when a big part of the DNA is missing. And here's an example of genes where chunks of DNA are missing. One gene, the Parkin gene, also causes Parkinson's disease, but causes dystonia in the feet, particularly at the start of the disease. And another gene called SGCE or epsilon sarcoglycan often has a missing chunk of the of the gene, and this is only picked up using special analysis techniques. So a mutation in this gene, SGCE, causes a condition called myoclonus dystonia. I'll show you a video of one of the patients diagnosed in the study to show you some key features of myoclonus dystonia. Myoclonus is this jerky, involuntary movement, and you can see the patient also has pulling back of the head, and he has a jest, which is touching his head. The jest relieves the dystonia.
What do? You touch your face and
Yeah, touch your face all the time.
what does that do?
It tries just some attempt to try and stop moving.
This
Yeah.
disorder, there's
You find
patients
it better.
improve if they have a drink of alcohol, so it's alcohol responsive. So we looked over our genome data and we tried to see, well, what is it? What features that may predict that a person is going to get a genetic diagnosis? And we found that patients who are young, who are young when their dystonia started, that's age at onset, who have combined dystonia, so when dystonia is combined with another movement disorder, or if they have uh, 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 segmental dystonia, they also are more likely to get a genetic diagnosis. In fact, if you have dystonia which affects only one body part, like just cervical dystonia and that's it, you're unlikely to get a genetic diagnosis. So we diagnosed 11 to 12% of patients, but what about the other patients? So that was the next study that we attempted. We had this hypothesis that purely by looking over the data again many years down the track, we can improve the diagnostic yield. And this is called reanalysis. So after doing the first study that did genome sequencing in dystonia, we then did the first study to reanalyze genomic data in dystonia. And we showed that the diagnostic rate increased by quite a bit. It increased from 11.7% to 18.9%. So a 7% increase in diagnostic rate. And you can see here all the new genes and the new diagnosis that we made. And I'd like to thank Dr. R.V. Fellner for undertaking this study. He worked with me as a visiting research scientist. Now, some of the new diagnoses were due to the discovery of new genes. And this is one of the major contributions our dystonia research study has made. We've contributed to the discovery of brand new genes that no one knew about. In some cases, we led those discoveries. So here's a new gene called VPS16. And in this patient, the dystonia involves involuntary movements around the mouth and tongue, oromandibular dystonia.
Go to sleep, it goes away.
Yeah.
Not just touchy.
This is a gene that I discovered with a colleague, Dr. Michael Zeck in Germany, and Sylvia Bosch. It's a gene called AOPEP. And you can see the patient has dystonia due to a mutation on both copies of the gene AOPEP. And this is a patient of Professor Victor Funks. And you can see that see her walking and she's holding her head as a jest to relieve her dystonia. So, after doing our reanalysis study, we again looked at what are the predictors that predicts that you'll get a genetic diagnosis in dystonia. We had more information now, and Dr. Michael Zeck had come up with a scoring system to predict whether you'll get a genetic diagnosis in dystonia. And so he allocates 2 points if you had dystonia that started in infancy or adolescence. He allocates 1 point if you had generalized or segmental dystonia. He allocates 1 point for combined dystonia or 2 points for dystonia, isolated or combined, plus non-motor movement disorder symptoms. And then we looked at that scoring system, and really we showed that if you don't score at least 1 point on this scale, there's virtually no chance of getting a genetic diagnosis. So there are different ways to think of who should get genetic testing in dystonia. And one way to look at it is this worthwhile? Are you likely to get a genetic diagnosis from the gene test? And you could use this scoring system. And if you get no points on it, you're probably not going to get a genetic diagnosis. There are other factors that you could consider. Does the person really want to have a genetic test? Do they want to know? And that is something we should consider. Or if a person is going for deep brain stimulation, because the type of gene that you have can predict your response from deep brain stimulation for dystonia. I'd like to move on now to a new study that we're doing. It's called a Functional Genomics Approach to Understanding Dystonia from Genetic Code to Gene Expression. And it's part of the Ainsworth for Dystonia Genetic Research Mission. So what are the aims of this study? Essentially, we're again, using advanced genomics to try and understand about dystonia. We're using RNA sequencing to try to increase genetic diagnosis and gain insights into the disease mechanisms. And we're using a new approach called long read sequencing to try to get an early and accurate diagnosis of dystonia. And basically, we're trying to improve the diagnostic yield. We got it up to 20%, but we want to get the diagnostic yield higher. What about the remaining 60 to 80% of patients who don't have a genetic diagnosis? How can we learn more about dystonia? And how can we give people a diagnosis and learn more about the disease and the disease mechanisms? So this research is going to provide a much needed capability to bridge the gap between clinical and research supported diagnoses and pave the way for introducing a more comprehensive diagnostic approach into clinical practice. What we want to do is boost the diagnostic rate, shorten the time it takes to get a diagnosis. This is called the diagnostic odyssey, and reduce the burden on patients from not getting a diagnosis, getting unnecessary treatments, getting unnecessary investigations, and try to improve patient outcomes. And I would like to thank the Dystonia Research Network for supporting this research. They helped in publicizing our research, and they've given us a large boost to recruitment. We've worked. One of the patients who took part in the research, which involves a skin biopsy, has written a nice description of the experience. If you want to read it, there's a link down below, and you can search it on Google, and you can find and learn about what it's like to come in to a hospital and have a research study like a skin biopsy. Because at first it sounds quite scary to have a skin biopsy, and hearing about another patient's experience can be helpful to understand what that experience is like. And here they've given a little description of, they've given me 5 minutes and a bit of a description about my research and also my interests. So the RNA sequencing study is doing RNA sequencing off skin fibroblasts. So why do we choose skin? Because we know that skin is similar to brain tissue, and many of the genes that are turned on in brain tissue are also turned on in skin tissue. So there's similarity between nerve tissue and skin tissue, and the skin biopsy is a useful repository of information. We can take a skin biopsy and store tissue and use it for other studies, like turning it into a stem cell and then a nerve cell. So it's a great resource that we're building, and in this study we're looking at sequencing the RNA. So you may wonder what RNA is. So if we look at it very simplistically, genetic code is made up of DNA, and DNA gets turned into RNA, which is like a messenger to carry instructions from DNA, and that is used to make proteins, and proteins are important for the functions in our cell. So if we look at gene expression, it can tell us the consequences of change in the DNA and help us understand the impact of DNA changes. So it gives us more information. And I'll like to give you an example of how RNA sequencing helped us solve a case, a very complex case of a patient who we couldn't find a diagnosis for. So here is this 27-year-old woman who has dystonia and also has nerve problems with her nerves called polyneuropathy. And she was being treated with drugs to treat the polyneuropathy, assuming that it was due to inflammation. She was treated with a drug called intravenous immunoglobulin, a drug called rituximab. She was getting complications from these treatments, such as infections. If you see a video here, you can see her dystonia.
Watch, look, if you check your eye movements, so keep your head still and watch my.
You see that she has these twitching movements of the face called dyskinesias.
Now, I want you to squeeze your eyes shut for me, thanks. Open your eyes. Show me your teeth. Hold your hands out for me, please.
You can see that she has dystonia in the hands as well.
Turn the hands over.
These involuntary postures of the hands.
Touch your ear and touch my finger.
Okay, and so basically we did that study that I was describing. So we took a 3 mm skin biopsy here. We grew it on a dish, and I did this with the help of scientists. And then by studying the RNA, we were able to show the patient has a mutation in a gene called IRF2BPL that's found that we identified from the RNA. So in this patient, the genetic diagnosis was made from RNA sequencing, allowing us to stop harmful, unnecessary treatments with drugs that suppress the immune system. And here we can all show this figure shows that we can also get additional information from the RNA sequencing data. We can look at gene expression patterns and compare patients who have dystonia and which genes are turned on and off, and compare that to patients who are healthy. And that gives us a clue. So maybe it tells us which pathways are activated by these abnormal dystonia genes. And it can also give us a clue, well, maybe a protective pathway is being activated to prevent the dystonia. So this really gives us clues. What are the pathways, the molecular pathways involved in dystonia? And hopefully these insights will one day lead to treatments, new treatments for these disorders. So our expected outcomes of this RNA sequencing study is to identify new genetic diagnosis. We can also look at how a gene is spliced. Splicing is how you go from DNA to RNA. You remove these non-coding regions of the genome. But if there's an error with splicing, the way the RNA is put together is abnormal. And this can be a clue to a genetic diagnosis. So we're going to try to find new diagnoses, and we're going to look at gene expression patterns. Another aspect of our dystonia research mission is looking at long-read sequencing. And this is something that I'm really excited about. The Garvan Institute is one of the world leading centers for long-read sequencing. And long-read sequencing is the third generation of sequencing. It's revolutionized how we do DNA sequencing. So before, we did short read sequencing. We chopped up DNA into small sections and then aligned it to a reference genome, and were able to identify spelling errors in the gene. But we weren't very accurate at detecting other types of mutations. For example, stutters in the DNA. For example, you can have 3 letters in the DNA, CAG, CAG, CAG. If those 3 letters are repeated too many times and it crosses a certain threshold, it can result in a disease. These diseases are called repeat expansion disorders. And it turns out that long read sequencing is particularly good at detecting repeat expansion disorders. And repeat expansion disorders can cause dystonia. They're particularly important for neurological diseases and also can cause dystonia. Long read sequencing can also tell us other changes to DNA, like methylation patterns. And methylation is really important for some genetic forms of dystonia, such as the gene KMT2B and the gene that I've already mentioned about SGCE. So in the past 12 months, we collected at least 40 blood samples for long-read sequencing. The long-read sequencing is now our preferred approach for DNA sequencing, and we're in a world-first approach. We're covering the entire DNA, the genome. We're doing long-read sequencing across the entire genome. And we are competing with some colleagues overseas. It's a race to see if we could be the first to publish a study on long-read sequencing in dystonia. This approach is really cutting edge, and I'm really excited about it. So we're doing long-read sequencing that covers the entire genome, and it will allow us to find all sorts of mutations. And here is a recent paper in this really important journal called Movement Disorders. We published a landmark review into long-read sequencing for dystonia. And there are 3 groups working on this across the world. And the leaders of each of those 3 groups, we got together and we wrote this review. So I want to show you an example of a patient who we diagnosed with long-read sequencing. You can see the patient has these jerky movements of the head. This is Professor Victor Fung examining the patient. So cervical dystonia, tremulous cervical dystonia.
Same with the left hand.
And you can see that she's of East Asian background.
Like this.
Good.
And now what the doctors noticed is that her MRI was not normal. The MRI has these changes in the white matter of the brain, and there's also these bright signals called called DWI, diffusion weighted imaging, shows bright signal. And then they did a skin biopsy and showed these inclusions. So this disease is called neuronal intranuclear inclusion disease, or NIID. And our approach using long-read sequencing, we showed that this disorder was due to a stutter or repeat expansion in the gene called Notch2-NLC. And there is no test for this in Australia outside of a research study. So we're the first really in Australia to be able to test for this in dystonia. So I'd like to acknowledge all the patients who provide samples and participate in our research. And I'd like to acknowledge the funding from the Paul Ainsworth Family Foundation, and to my team. And we've got a team of Karen, who's a project manager, PhD students Dennis Yao and Laura Rudax, and a postdoc, Sue Fei Xiao, and other grant bodies, and all our collaborators at Westmead, St. Vincent's, Neura, and our international collaborators, including one of my mentors, Professor Christine Klein. And if you're interested in finding out about this research, or you have any questions, I'd be really happy to answer any of your questions. And you can see my email here, and I'll be happy to take any emails. So thank you very much, and I thank you again for Dystonia Network Australia.